Protein synthesis, also known as central dogma, is the process in which an organism produces proteins. To better understand how it works, we must first know what is involved. Atoms are particles of chemical elements that, when put together, create chemicals called molecules. The smallest molecules are called monomers, and they come together to create slightly larger molecules called polymers. Polymers go on to create bigger biomolecules, or molecules produced in an organism, which go on to create even larger macromolecules. The initial monomers include monosaccharides, which create the carbohydrate polymers; fatty acids, which form lipid polymers; nucleotides, which make nucleic acid polymers; and amino acids, which produce protein polymers. Protein synthesis focuses on this last one, amino acids creating proteins. To begin, we have deoxyribonucleic acid, abbreviated to DNA, which is genetic information found within the nucleus of cells that help organisms develop, live, and reproduce. DNA is made of nucleotides, which are compounds created from a sugar, a phosphate group, and a nitrogenous base. The nitrogenous bases in DNA are adenine, thymine, guanine, and cytosine. The sugar in DNA is deoxyribose, which is made from ribose, a natural compound generated through the consumption of food, and the molecule, hydrogen. DNA is formed into the shape of a double helix, which looks like a twisted ladder. The backbone of the double helix contains the deoxyribose and phosphate group, while the nitrogenous bases are paired with their complement on the inside. Adenine pairs with thymine, while guanine matches with cytosine. When a certain protein is needed, the body sends a signal to the cell so it can activate that protein's gene. Genes are sequences of nucleotides that have the potential to create a protein. This is the beginning of protein synthesis. This also releases the enzyme, RNA polymerase, onto the DNA strand. The RNA polymerase makes its way across the split gene, pairing the complementary nitrogenous bases to the original DNA bases. Except, instead of thymine being paired to adenine, the base, uracil, is added. This creates RNA, a single-stranded sequence of bases and their phosphate and sugar groups. The process of creating RNA off a DNA template is called transcription. The RNA is now able to leave the nucleus as mRNA, or messenger RNA. It travels out of the pores of the nuclear membrane and into the cytoplasm, or the jelly-like fluid in the cell. Here, ribosomes attach to a specific codon, or group of 3 nitrogenous bases. The starting codon is AUG, which codes for the amino acid methionine. The ribosomes continue reading this sequence of bases, called a reading sequence, producing more amino acids in the process. The amino acids are carried into a sequence of amino acids called a polypeptide chain, that is attached to another type of RNA called transfer RNA, also known as tRNA. tRNA has the anticodons, or complementary base codons to the mRNA sequence. This second process is called translation. The ribosomes finish at one of the 3 stop codons, UAG, UAA, or UGA. When the order is completed, it folds into a 3-dimensional figure, which becomes a functional protein. Proteins are polymers organisms need to function, maintain homeostasis, and repair their structure. Now, what happens when something goes wrong? A mutation will occur. A mutation is a flaw or flaws in the genetic code of an organism. There are 3 mutations: insertion, deletion, and substitution. Insertion is when a base, or multiple bases are added into a DNA strand. This is an example of a frameshift mutation, in which the rest of the codons will be affected. Another frameshift mutation is deletion, in which a nitrogenous base or multiple bases are deleted from a DNA sequence. The final mutation, a substitution, is when one base is replaced for another. This is an example of a point mutation, in which only one base pair, and ultimately one codon, is changed. These mutations not only affect which amino acids are created, but also how many. If a base or bases are altered, then the start and stop codons may be different. If there is no start codon, then the ribosome will either attach to the next available AUG codon, or not create the proteins at all. If the stop codon is altered, then the ribosomes will either end at the next available stop codon, or continue producing amino acids until the reading frame is completely finished being translated. The overproduction or underproduction of amino acids will either change the function of the protein created, or not make the protein function at all. Sometimes, there are leftover nitrogenous bases in a DNA or RNA sequence. These bases are utilized later to create more proteins. The process of protein synthesis seems very complex, but to simply put it, DNA is turned into the single-stranded RNA during transcription. Then, the RNA is transformed into amino acids during translation. The amino acids form proteins that help the organism function. Sometimes, there are mutations that can cause the protein to malfunction or not work, and extra nitrogenous bases that will be used later.